Hello, everyone, and a warm welcome to each of you who has joined us here today. I am Bruna Lousada, the European Manager of Teledyne Hansen, and it brings me great pleasure to introduce this enlightening webinar, Using Human Skin in Permeation Studies, Ethics, Regulations, and Models, with Dr. Laetitia Rapetti, the Chief Scientific Officer from Alphenyx. Alphenyx is a French company specialized in sourcing and transformation of human tissues, such as the dermatome skin discs we used in permeation studies. Their mission is to provide alternative solutions to animal testing in research. Dr. Rapetti's academic journey is truly impressive. She graduated from the prestigious École Normale Supérieure Paris-Saclay. Following that, she pursued studies in immunology at the Pasteur Institute and earned a PhD in physiology and cell biology with great honors from Paris 5 University. She has then spent 5 years as a research associate at the University College London in the UK and currently collaborates with Professor Philippe Picherellis at Aix-Marseille University on some groundbreaking research projects focused on skin autoimmunity. So last year, which is when I met Laetitia for the first time, we started a collaboration between Teledyne Hansen and Alphenyx, combining our advanced diffusion cell instruments with Alphenyx high-quality skin models for penetration studies. So this partnership aims to provide enhanced value, cost efficiency, and a holistic solution to all the in vitro permeation testing community. So before we dive into today's presentation, I want to remind you that all attendees are currently on mute, but you can submit your questions at any time using the Q&A section at the bottom of your screen. We will make every effort to address all your questions at the conclusion of the presentation, but if it happened to run out of time, rest assured that we will follow up with you directly via email. If you experience any issues with your audio or connection, we recommend logging off and then logging back on. For those who can't watch the entire webinar or wish to revisit later, we will be making it available for replay on the Teledyne Hansen YouTube channel in the coming days. With that said, I'm delighted to welcome Dr. Laetitia Rapetti to begin our presentation.
Hi everyone, and thanks Bruna for this introduction. Before discussing our topic today, I will first give you some information about Alphenyx. We provide alternative solutions to animal testing for fundamental research, but also preclinical studies of new drugs and safety, efficacy evaluation of chemicals, cosmetics, indeed any molecules intended for human use in general. We are therefore specialized in human skin models for permeation studies. When working with human biological samples, high ethical and regulatory standards are obviously mandatory because it involves our legal responsibilities as a supplier, but also our users' responsibility. The main point about ethics and regulations is that they may differ from one country to another. So it is advised to sign a material transfer agreement or a master service agreement with suppliers to define in particular applicable ethics and regulations and what we are allowed to do or not with the human biological samples and the related clinical data. In terms of ethics, human skin and its derivative models are classified as HBS, human biological samples. They are part of the human body. Therefore, ethics to respect donors' rights are essential and intrinsically linked to laws and regulations. Before collecting human samples for research, an institutional review board or an independent ethical committee approval is mandatory. Most suppliers already have an ethical approval in place, but not always. So if it is not the case, the user must obtain an ethical approval for his study. The Declaration of Helsinki, which outlines the ethical principles about research on human subjects for clinical trials or any research directly involving a human being, is a good reference. Although skin for penetration studies is a discarded tissue and not part of a human body anymore, following the standards defined by this declaration is recommended. In France, the French Ethical Committee is named the CPP, Committee of Person Protection. This committee is in charge of approving research involving human beings. As a supplier, or authorizations for collection, preparation, use, storage, and transfer of human origin tissues for research use only have been reviewed and approved by the CPP. The CPP has therefore approved the procedure established for obtaining donors' consent and sourcing human biological samples. In most developed countries, there is an ethical consensus. Each donation must be voluntary and anonymous. To respect the donor's will, the informed consent form is crucial. All donors, or family in case of a deceased donor, must have been duly informed. An information letter must meet specific points according to local regulation and be as specific as possible regarding the use of the samples. All donors or family have therefore signed an ICF before collecting the samples. Another point, which is really important, at least in France, the donor can revoke its consent at any time before or after the gift. Any remaining HBS materials must be destroyed immediately, but research data generated with the materials before the date of consent retrieval remains legally exploitable. In addition, to respect donor privacy, access to the signed ICF is strictly regulated and limited to the clinical staff. Regarding genetic studies, they are possible if the donor gave a specific consent for this type of studies, but it is strictly regulated, as we will see in the regulation section. In any case, information regarding genetic studies must be mentioned in the document provided by the supplier. The second important point about ethics is that the donation must be anonymous. To respect donors' rights in France, only the medical staff has access to donor identity. Under no circumstances, donors' names or any personal identifier could be provided. We follow ourselves a double-day identification procedure, ensuring donors' full privacy and sample traceability. In addition, the clinical data provided must be treated with the respect of donor privacy in compliance with general data protection regulation. Indeed, ethics is closely linked to regulation. So we are going to move on to our next part, which is regulation. As I said in the introduction, regulations on HBS depend on your country. In France, it is defined by the Code de la Santé Publique, in the US by the FDA, in the UK by the UK Human Tissue Act, by example. Before using human skin in your lab, you must meet specific legal requirements as a user. You must yourself comply with your local regulations and possess the required authorizations to use human samples for research delivered by your country competent authority. It is also advisable, due to the differences between countries, to verify that your supplier is fully authorized for its activity in your country and that the samples meet your local ethic regulation requirements, and also, if you have one in your company, your internal code of conduct. As sometimes in big companies, they have internal standards even more restrictive than local laws. To work with HPS, you must consider three aspects of regulations. Regulation on HPS by itself, including importation and exportation, but also regulations on personal data collection for the data collected from the patient, and finally, transport regulations. To start with regulation on HPS for research use, there are two main principles shared in developed countries. The donation must be anonymous to respect donors' privacy, and voluntary. Voluntary means duly informed using an ICF as we saw in the ethic part, and with no contraint or whatsoever. There are some differences regarding consent for deceased donors. Some countries request an explicit consent, like in Australia, Brazil, Canada, Germany, or the United States. Other only needs a presumed consent, like in Argentina, Belgium, Spain, or Japan. In addition, the donation can be free or remunerated depending countries. In France and most European countries, donation is considered as a gift, so it is free. But in other countries, like in the US, it could be remunerated. Be aware, however, that some big organizations' internal charts may require a free donation of HPS for research use. In Europe, suppliers comply with the Council of Europe regulation for protection of human rights and dignity of the human being, as defined by the Convention of Human Rights and Biomedicine. Use of HPS for research is regulated in particular by the 2004-23 CE directive. In France, it is also regulated by specific articles from the French Public Health Code. Of note, French regulation is one of the most stringent, so French suppliers are compliant with European law, obviously, but also the FDA, UK law, Swiss law, and many other countries. In France, there is a principle of non-commercialization of the human body or any part of it, so the donor perceives no payment for a surgical discarded tissue, as discussed previously, and it is also the case for the clinician and the clinical center. There is no direct remuneration for the act of tissue removal by itself, but compensation for their time and costs related to donor information, selecting the donor, handling samples and clinical data, and so on. Regarding the final user, the payment to the supplier is for the service linked to the HPS processing, preparation, and associated clinical data handling, and not the purchasing of the human samples. So be aware to not use the word samples were purchased, especially in publications. In addition, in France, there is an obligation of traceability of the clinical data and donors record for 10 years. Finally, on safety issue, all donors are tested negative for the presence of HIV, hepatitis B and C viruses. To give you an example about on authorization as a supplier, the authorizations for the use of HPS from living donors are delivered by the French Ministry of Research with the Committee of Person Protection approval. There are three types of authorizations: the DC types, which are mandatory for any users of HPS, the AC types, which is specific for suppliers. It allows them to provide HPS to users for research, and the IE types to import HPS from abroad and export them to foreign users. Indeed, any non-French user must be declared to the Ministry of Research. These two last types of authorizations have been to be renewed every 5 years. In addition, if you collect samples from deceased donors for research use, you also have to obtain a specific authorization from the Biomedical Agency. So we are going to move on the second part of regulation, which is general data protection regulation to protect personal data from the donors. Be careful, as GDPR rules depend on the country of origin of the HBS. In France, GDPR is defined by the EU Parliament and is controlled by the CNIL, which is a French national organization for GDPR. Technical donor data provided by the supplier has to be treated by the user with respect of donor privacy and with the same level of protection as that of the HBS country of origin. GDPR is harmonized for all EU members, and the same level of protection is applied in many other countries like Switzerland, the UK, and Japan. However, for the US, by example, it is less restrictive. As a general rule in the European Union, suppliers must perform a risk assessment in order to prove that the risk of re-identification of the donor is negligible to respect donor anonymity. The user must comply with its local GDPR and protect the transferred data and the data generated with the HBS by proper security measures. For example, only data useful for the study must be transferred, and only people involved in the study should have access to this information. As a consequence for genetic studies, they must not increase the degree of risk of re-identification of the donor. In any case, genetic studies which are likely to breach the donor's anonymity are strictly forbidden. Our last point today about regulation is transport regulated by the International Air Transport Association. Even if they are tested negative for HIV, hepatitis B and C viruses, HBS are classified as dangerous goods and are submitted to specific rules for transport. They must be labeled as UN 3373, meaning biological substance category B class 6.2, and with a specific UN 1845 label for dry ice for shipping frozen samples. The packaging must respond to the P650 norm, which is basically a triple packaging protection. Be aware that only accredited transporters can handle this kind of products. To give you an idea of delivery times from France, HPS can be delivered within less than 24 hours for most European countries, 2 to 3 days for America, and 3 to 5 days for Asia. We are now moving to the last part of this presentation. As we saw, there are a lot of constraints regarding ethics and regulations for the use of HBS. So the question is, why using human skin as a model for permeation studies? Indeed, the current regulations in developed countries impose strict controls on any product for human use that are potentially dangerous for human health, like drugs, cosmetics, but also food additives and other chemicals. Efficacy must also be proven before obtaining a marketing authorization. Available models for permeation studies are animal skin, synthetic membrane, and human skin. Until recently, this test involved the use of thousands of animals. There are lots of in vivo and in vitro models to perform permeation studies according to OECD, like rabbit, rat, hairless mouse, even dog or monkey. Recently, scientists around the world became more conscious about animal welfare, and authorities from most developed countries aim to move towards a more ethical research. There are new regulations in the European Union and in the US in particular, with the 3R principle, which is reduction, refinement, and replacement of animal experiments in research when another equivalent in vitro method exists. The ban on animal experiments to test cosmetic products. Any new cosmetic tested on animals won't obtain a marketing authorization in European Union, but neither in the US. The REACH guidelines on chemical products and the OECD guidelines for the use of alternative testing methods. There is, however, an exception, an ethical exception regarding the in vitro pig skin model, because pigs are not killed for research, but for human food. It is far cheaper and easier to obtain compared to human skin because there is almost no regulation. But you must be vigilant with the quality of the samples, including the conservation temperature and the time elapsed since the samples has been collected, maximum 24 hours after pig death. The sooner the better. You can obtain ear skin from slaughterhouse, which is better because the thickness is closer to human skin. You can also obtain other skin parts from butchers directly, but be aware the skin is really thick and it is not easy to have an even and reproducible thickness when you dermatose the skin. According to OECD, skin pig is the most representative of animal skins compared to human skin. If the quality of the sample is good, you can obtain quite consistent results compared to fresh human skin. However, some significant differences have been documented recently in publications, and it is hardly predictable. So pig skin is a good model for first screening, but at some point, if there is no information in the publications for your formulation, it is better to confirm the results on real human skin. In any case, you can't use frozen pig skin for permeation studies, as there is a well-documented increased permeability compared to the human skin. Synthetic membranes are also a good model. They could be a cellulose membrane or have a more complex composition like the Strat M, which is the most commonly used in the US. They are more or less economical depending on models and providers. They are commonly used in the US, less in Europe, because certainly it is easier to obtain real skin in Europe. A good point is that there is no problem of conservation or availability, and you can obtain consistent results with minimal low to low variability. However, again, it is useful for large screening, but it won't reflect real skin complexity and individual diversity. In addition, percutaneous absorption studies aim to estimate the passage of a substance across the skin and assess the proportion of the substance in the different layers of the skin: stratum corneum, variable epidermis, dermis, and bloodstream. With synthetic membranes, you can't assess compounds biodistribution in the different skin layers. Moreover, you can't perform metabolism studies, which could be important because your compound can be metabolized by the skin and become a different product more toxic for the human organism, by example. Regarding these limitations, real skin models are always preferable if possible. For instance, reconstructed skin models are not recommended by OECD because their skin barrier properties are weaker than fresh or frozen human skin, but they could be a good alternative in the future. Human real skin is generally considered as a gold standard and preferable for permeation studies because the tested molecules are usually intended for applications to the human population, and there is a high correlation in the literature between in vitro and in vivo data. It reflects real skin complexity, but also donor diversity. So due to inter-donor viability inherent in biological membranes, human skin from different donors should be included in your study. And according to OECD, more donors and replications might be required for bioequivalence assessment of topical products than for risk assessments. You can use skin from living donors. In that case, skin tissues are collected from patients undergoing plastic or reconstructive surgeries. Alternatively, cadaver skin, which is a non-viable but intact skin, could be used according to OECD. Cadaver skin is not really used in Europe for ethical reasons mainly, but more used in the US because it could be easier to obtain than skin from living donors. From living donors, you can either use abdominal or breast samples for your studies. According to OECD, you can use either fresh or frozen skin because the skin barrier function is not affected by freezing. However, you must avoid repetitive cycles of freezing and thawing. Obviously, you must use fresh skin to perform metabolism studies. You can also use either full thickness skin, up to 1 millimeter thickness maximum, or dermatom skin. It is, however, recommended to use dermatom skin between 200 to 400 micrometer or around 500 micrometer. To summarize, suppliers can provide, depending on your study design, fresh or frozen skin, full thickness or dermatom skin, or even ready-to-use dermatom skin discs, which is easiest when you are not used to dermatom yourself. In any case, to obtain consistent results, you have to use good quality samples. The quality of the skin depends on its conservation. The maximum viability and quality is obtained if the skin is conserved at 4 degrees as soon as possible after skin removal from the donor. The length time between skin removal and its use as fresh samples has to be kept to a minimum. It is recommended to start the experiment within 24 hours max after skin removal for fresh samples, and to freeze samples as soon as possible and preferably within 12 hours or at the maximum within 24 hours after skin removal. The storage conditions for frozen skin are at -20 because at -80 storage, it affects the skin barrier, and it is recommended to avoid contact with air to limit oxidation. If stored in good conditions, human skin can be stored at -20 for several months, up to 1 year, without affecting the barrier function. Finally, as it is mentioned by OECD, the thickness homogeneity of the dermatom skin disc are also important. Before using the samples, it is therefore recommended to test its quality, its integrity with the transepidermal water loss test, its viability if you use fresh skin with a glucose or a MTT test, and at last, its thickness. It is agreed that the dermatome allows to set a specific thickness, although the skin is not always cut evenly. It is well documented that this can induce variability. So a good homogeneity is very important. Most researchers use a micrometer or the Audi test to control thickness. The number of points measured is, however, limited to 5, 6, or 10 points. The guidelines underline the necessity to have homogeneous thickness, but don't define the number of points to measure and the spatial distribution to guarantee reproducible results. To conclude about thickness measurement, obviously it must be controlled for each skin disc individual. In addition, to assess homogeneity, we use at Alphenyx an innovative system for higher sample reproducibility. This system allows us to brush the surface of the skin disc and measure over 100 points per disc in real time. We can then therefore obtain, using a specific software, the mean, the minimum, and the maximum skin thickness, which allow us to define reproducible quality standards for the skin disc. In conclusion of this webinar, using human skin in permeation studies must meet specific legal requirements. Ethics and regulations differ from one country to another. Users and suppliers must be compliant with their local laws. The MTA or MSE must be established for the use of the HPS and associated clinical data to protect both parties and apply the highest standards. Using human skin in permeation studies is the gold standard according to OECD, but samples availability can still be tricky in certain countries. Thank you for your interest. If you have any questions, Bruna, maybe.
Thank you, Laetitia, for this excellent presentation. So we will now move on to the Q&A session. Just a friendly reminder here that you can share your questions by clicking on the Q&A button situated at the bottom of your screen. Okay, so here is the first question for you, Laetitia. EMA draft guidance on quality and equivalence of topical products states that it is recommended to use ex vivo adult human skin. It does not specify if it's surgically obtained from abdominal or breast surgeries, and it's acceptable. Do you know if ex vivo human skin from cadavers can be used in IVPT studies and submitted to EMA? Because in the US, the FDA accepts both sources, surgical and cadaver, with no preferences.
Okay, yes, like the FDA, EMA accepts both skin from living donors and from deceased donors, as soon as you can guarantee that the quality of the samples is good. So you have to assess it, first of all. As I say in my talk, different parameters are really important to guarantee the skin viability and quality. Like, by example, the time between the skin removal and its use for research. And samples must be refrigerated at 4 degrees as soon as possible, which could be a little bit more tricky for deceased donors than for living donors. To our experience, however, most of big organizations, open organizations, prefer to use only skin from living donor. I don't know really if it's because an ethical reason or an internal rule of the company,
Mhm.
or if it's because it's easier to have a better quality of the skin using living donor.
Mhm. And that's mostly your experience in Europe, correct?
Yeah,
Yeah.
exactly.
Thank you, Laetitia. Next question. It's actually asking if you could talk a little bit more about the thickness measuring tool. Do you have any other specific information about that tool, or can you share more?
You have more information in the OCG guidelines. I can share the publication with you. As I say, the OCDs say it's really important to control the thickness, but it doesn't specify the number of points you have to measure and the repartition of the points of the skin surface. Most people are using a Faber system or something like a laser system, and usually they are measuring something like 3 up to 12 points, to my knowledge. For us, it was really important to better control this thickness. That's why we put in place a new method to assess it, but it's a trade secret, so we won't share our method because it's our trademark.
Okay, of course. It's a proprietary technology, I would say, correct?
Yeah.
Very good. Another question about the US again. So how does custom clearance of those HBS works in the US? Do you have any experience?
So we already have a couple of users in the US. So for instance, we have no problem with custom clearance. Sometimes, so as I said, from France to the US, it's 2 days, up to 3 days. It depends if you are located on the east or on the west coast. But I mean, we already have 1 day delays from custom. But the good thing with FedEx is that they are keeping the samples at -20 for frozen samples, and they are able to adapt dry ice, so the samples can be kept frozen. So there is no problem when you receive them; it's still frozen, and you can use them for your penetration studies.
Very good, thanks, Laetitia. Next question. What is the reason for storing at -20 degrees C rather than 80?
Okay, it's really important to store the skin at -20. There is several publications showing that at -80, sorry, the skin barrier is affected. So it will damage the skin and you will have a higher permeability.
Very good. Next question. Is there any living skin equivalent available that mimic cadaver skin, or I guess human skin?
Not to my knowledge. You mean like membrane, like synthetic membrane? No.
If it's a synthetic, I don't know. It doesn't say.
Because there is Strat M membrane, and there is a couple of articles comparing the Strat M to real skin. So I can share the publication as well. It's comparable, that there is some differences. We submitted, I mean, we got an article just approved today to the International Journal of Pharmaceutics, where we are showing that Strat M is acceptable, equivalent to human skin, but it's not perfect. So I can send you the reference as well if needed.
That'd be
I
perfect.
don't know if I answer the question.
Okay, yeah, we can share that with the audience. That would be perfect. Next question. Is the skin from surgery better than cadaver skin? What about the metabolism in this skin?
The thing is, the crucial point is really for cadaver skin, if you remove the skin immediately, you can have a comparable quality to living donors. I'm not really used to cadaver skin, honestly, because we don't use it at Alphenyx for this reason, because I think it would be really tricky to remove the skin from a deceased donor very quickly after the death, for ethical reason, obviously. From what I saw in the publication is for skin grafts on real human. The graft is working better if you remove the skin at least 24 hours after the skin death, and you have to keep it really at 4 degrees. Because if you, after 24 hours delay, the graft is not, it's not working.
Okay.
So yeah, the problem with cadaver skin is to guarantee the quality of the skin because of logistical organization. Because from living donor, it's quite easy to take care of the skin and using the skin in research. I mean, as soon as possible. In our lab, by examples, when the skin is removed from the donor, we receive it at our lab in something like half an hour or an hour before skin removal, which is quite fresh.
Very good. So it's mostly for the logistics, the problems with the cadaver and quality, as you said. Yeah. We have many questions. Another question, as the skin thickness differs at different sites of the body, does Emma has any recommendation regarding the sample site for topical and transdermal permeation studies?
To my knowledge, there is no, I think abdominal skin is recommended, but you can also use breast skin. It's true that the skin thickness depends from one location to another, and also from one donor to another. It depends the BMI of the donor as well, because sometimes the skin is more stretched than other times, and the dermis could be really thin or really spongious, so it's really donors dependent. The only thing is when you dermatom, it is recommended to dermatom either from 200 to 400 micrometer or around 500 micrometer. In this way, if you use 500 micrometer, you have something like 200 micrometer of epidermis, roughly, and 300 micrometers of dermis, which is a good representation of the skin.
Perfect. Next question. How increased is the permeability when frozen pig skin instead of fresh pig skin? So you did mention the permeability issues.
Yeah, I don't have the numbers in mind, but I have the publication. So there is, to my knowledge, 2 or 3 different articles I've read recently, and a review also stating that frozen skin pig skin is not increase the permeability, and I can send the reference to the person who asked the questions.
Very good. So we will send you via email all of those studies that Laetitia is mentioning. Very good. One last question. It's actually from asking about the use of skin on IVPT studies in China. Do you know how that works? I've heard recently that it is prohibited. I don't know. Do you have any information?
So I don't know really if it's prohibited in China. So what I know is that in any case, a foreign supplier can send and ship human skin in China because they are not accepting dangerous good, and skin is classified as UN 3373 dangerous good. So it won't pass the custom clearance. So any foreign suppliers can send human samples to China for importation, but I don't know if inside China they can use human skin, collecting the skin themselves to hospital or Chinese supplier. I don't know.
Okay, thanks, Laetitia. And I think one last question before the end of our webinar. So again, if you have any further questions, please use the Q&A button. What is the difference between using split thickness and full thickness skin on IVPT studies?
The thing is, with full thickness, the thickness is not homogeneous. It depends donor as well. When you dermatom the skin in this way, you can homogenize the thickness of the skin from one donor to another, and from one disc to another from the same donor. In any case, the recommendation guidelines say if you use a full thickness skin, the maximum thickness must be 1000 micrometer at the maximum.
Okay, very good. So I think we don't have any other questions. So that's the end of today's webinar. Laetitia, thank you so much for sharing your knowledge and your expertise with us. And of course, thank you to all of you who participated and asked so many insightful questions. So as I mentioned earlier, this webinar will be on the Teledyne Hansen YouTube channel within a few days, probably before the end of the week. You can already check it out. You can watch this on demand and feel free to share with people that you think could also benefit from this content. And for more information on this webinar or to access any other Teledyne Hansen content or products, you can visit our website at teledynehansen.com or find us on social media. So thank you very much, and I hope to see you in our next webinar. Thank you.